Running. Good evening, Pittsburgh and universe. This is JC on a bike. I wanted to do just a great big shout out to the latest revolution in DNA technology, which was reported in the BBC a couple days ago and was released on, I believe, on BioArchive, or sorry, it was on Nature. And it comes from the Broad Institute, which is an MIT Institute collective with Harvard, and the Liu Lab, Liu is his last name, and I apologize for pronouncing it incorrectly. And what they've done is sort of take the CRISPR system, which was all the rage a year ago, and improved the way that it functions, the way that it works, to greatly increase the usefulness of the editing that they can do. And they now call it prime editing. And the idea is that they can target really very specific places in the genome. Now, let's take a caveat here and say that I'm not a molecular biologist, so all of you viewers who are, feel free to chime in the comments and correct any mistakes that I make here. But the gist of the CRISPR system is that it makes a double-stranded break and then inserts what it needs to insert. But the double-stranded break can also be a lot of other things to happen, and that includes a double-stranded break where you get insertions and deletions that you don't want. And those insertions and deletions that you don't want are a real problem in CRISPR that this new lab has really been able to solve, because instead of only being able to do 4-point mutations out of 12, for example, now they can do all 12. And instead of being able to fix a small sequence within the genome, they are able to fix very, very, very tiny sequences and replace them with the right ones. And these kinds of small changes, single amino acid changes, are very difficult to accomplish with CRISPR. Now remember that CRISPR was cool because it allowed a highly specific targeting of a specific gene, which gave us the ability to perturb genetic systems and molecular genetic machinery in a way that we haven't been able to do before, with much more specificity than we've ever had before. But this came at a price, in the sense that it wasn't perfect. And a lot of the mechanisms by which we manipulate DNA are not perfect, but instead rely on the high fidelity repair machinery that's present in all eukaryotic cells. That's sort of the trick. And that trick is sort of exploited on every in every way that DNA is manipulated. We rely on the high fidelity of the enzymes themselves to fix the strand breaks that we make, and during the repair we trick these enzymes or set them up to have a propensity to make the edit that we want. Now, historically speaking, that's been pretty difficult, and so to target a particular place in the genome has always been a challenge. I mean, you know, it wasn't too long ago that even finding the sequence you wanted would have been a challenge. So we have come a long, long ways. But the CRISPR system, again, I'm trying to explain, has this handiness, which is that you can target almost any gene you want pretty specifically, but it lacks the specificity necessary to address many of the known genetic disorders that require very specific editing at very specific sites. And the reason why the CRISPR-9 system doesn't work that well on those things is because, in those scenarios, is because the break in the DNA that the CRISPR system exploits is a double-stranded break. And a double-stranded break requires repair at two ends. And so a double-stranded break has 4 open ends, which really provides 4 possible non-desirable changes to be made, right? And so the open end of CRISPR is that for all the good mutations, you get a lot of ones you didn't want. A lot of them you didn't intend or didn't want. And so that sets up a scenario, or not sets up a scenario, but it sort of makes CRISPR limited in terms of what you can do for genomic DNA. Because certainly if you're going to mess with a human, you can't have a bunch of DNA that's wrong. Just have, you know, wait for the good one to show up. Holy festival of birds, Batman. He runs red lights with his music on. He runs red lights with his music on. And he rides no-handed. Places where people could open doors and knock him down. So he's riding with music, and he's riding no-handed. It's impressive. I'm surprised that he's still doing it, though, and living. So, double-stranded breaks in CRISPR means that you can target things and target very specific locations, but by the nature of the beast, you get a lot of errors, insertions and deletions that are unwanted and unavoidable. And so that means that if you want to, for example, change the one amino acid that's involved in sickle cell anemia, CRISPR is not the way to do it. If you want to change the 4 amino acids that need to be changed for Tay-Sachs disease, I think, CRISPR is not the way to do it. And it's not only because of the double-stranded breaking, it's because of the fact that the limitations of the system make it very difficult to do single point mutations to particular bases. And to correct many of the genetic disorders that we are familiar with in human biology, it's going to require just a few amino acids to change. And that means we're going to need something better than CRISPR in the future if we want to do things like that. And that's what this lab at MIT has managed to accomplish in this paper. Is they've taken the CRISPR-9 system and they've added some components to it to make it so that instead of operating on a double-stranded break, it operates on a single-stranded break. And more importantly, they've managed to incorporate a targeting sequence that can highly accurately nick the DNA in the exact place that the researchers intend. And the single-stranded break then is what results immediately in far fewer errors. And I'm sure there's a better and more detailed explanation for this, but from a cursory molecular biology perspective, it's important to understand this simple fact. DNA has two strands, and from each strand you can make a copy of the other, such that DNA is replicated by opening these two strands, separating them from one another, and then filling in the blanks on each strand. That's a normal thing. It happens all the time, and those are the exact enzymes that we want to hijack whenever we nick the DNA to try and make a change that we want. And in this case now, they just have created essentially a molecular targeting mechanism that allows them to look for specific sequences and put the enzymes there. And then again, you will see there's an equilibrium between the nicked DNA and its repair, and it's this equilibrium that they have biased toward the repair that they want, and biased it better than it is biased in CRISPR-9. Let's say it like that. It's still biased toward repairing back to the old sequence of DNA, so that you don't get a perfect change, but it does work. And more specifically, the number of errors has gone way down, and that's a really spectacular result. And I think the main argument that I would make in terms of citing this research is just that this is what will end up being the first step in terms of getting us to the stage where a lot of these genetic diseases can be addressed and maybe eliminated. And that's a pretty interesting stage of biology to be in, going right there. Because when I was a child, we were not even close to there yet. And a lot of these technologies were, we understood cartoon-wise what needed to happen, but the concept that we would have the knowledge and the skill to create an enzymatic complement that can do what we are doing in this paper. Thank you. And so in the link in the description, I'll put the paper, the link to Nature, and also the BBC article that might be better at describing the advance than what I've just done, but I hope this ride will at least serve to cue up the article for you and get you reading about it. I'm just amazed how fast science is moving now. It's even when I first got in, say 10 years ago or 15 years ago, science was moving slower, noticeably slower than it is now. All fields. It's because the internet allows us to share data and ideas in a way that makes it even harder for bad ideas to last very long. And that's pretty spectacular. But it also makes it very hard to stay relevant as a scientist. Because it only takes a couple years and everything that you wanted to research may be done, may be described. And that's what I think is interesting about old neuroscientists and young neuroscientists, is the old ones, they don't tend to change fields. They don't even change subject. You know, like the details, it's all the same. And they've been doing that since they were postdocs. Come on, guys. Come on, guys. Everybody's got a gas pedal. Let's go. Holy cow. It's incredible. Anyway, that was Journal Club on a Bike tonight. I'll try and wrap it up at home, I think. See why I have the whistle? Look at that deer right there. 2 of them. They just go away if I honk it. It's much better than having a deer at the side of the road and dinging with my freaking bell. Or yelling at them, which I used to do. Hey deer, get out of the way! That don't work. But my little screamer does. Hey, thanks for joining me on my ride again. Today we talked about an advance which was reported in BBC and in Nature magazine this week. It is an advance on the CRISPR-9 system which improves its fidelity and makes it possible to make all 12 point mutations as well as small group changes at specific sites in the DNA like never before. The term they're using for it is prime editing. It appears to be a major advance in terms of our ability to manipulate the genetic code in a specific manner. I relayed in the ride that CRISPR-9 was very good at targeting specific locations and specific genes, but not very good at the fine editing that is required to do a lot of the things that would be necessary if we wanted to fix human diseases where there's a single base pair or some insertion or deletion that's quite small. The best way to explain it is that you have a 2-stranded DNA and CRISPR makes a double-stranded break, so there are 4 places where errors can occur. And in this system, they've now gotten the CRISPR system to work on a single-stranded break so that it greatly reduces the possibility of error. And the accuracy of this nick has also gone up in the sense that they can nick where they want, regardless of what base combinations they're targeting. Again, I'm not a molecular biologist, so take all of this with a grain of salt. I'm only really telling you that there's something new out there and you should check it out. And that's what I want to do with these rides, is show you something new and let you check it out afterward. And I hope that I can start getting a daily ride together where I'm just popping papers out there for you guys to check out later. If you have suggestions for papers, please put them in the comments. And if you like and subscribe to JC on a Bike, and I'll see you next time.